Hello, I am Shi Qi and I am a biopharma scientist at Rocket Asia Pacific Innovation Center. Today I'm going to share with you how the use of hydroxypropyl beta-cyclodextrin can be used to extend the shelf life of therapeutic proteins, and also the mechanistic understanding of its stabilizations using bevacizumab as the model protein for our research work. Like any protein, monoclonal antibodies have the propensity to aggregate under a variety of environmental conditions. As such, protein aggregation is recognized as the key quality attribute of biotherapeutics, as it will reduce drug efficacy and may also potentially induce immunogenicity in patients. Monoclonal antibodies undergo aggregation via different pathways, such as partial unfolding of the native protein during storage, or via reversible self-association of the native protein to form oligomers. As such, formulation development is critical in identifying optimal pH and the right excipient that can help to limit the progressions of these aggregation pathways. In this work, we study how the use of hydroxypropyl beta-cyclodextrin, or also known as HPBCD, can be used as such an excipient. So what are hydroxypropyl beta-cyclodextrins? The native cyclodextrins, CDs, are cyclic oligosaccharides obtained from starch by enzymatic cyclizations and can be substituted with different groups, and in this case, a hydroxypropyl group. CD and their derivative have a bucket-like structure. It has a hydrophobic internal cavity which can allow complexation of small hydrophobic molecule, and a hydrophilic exterior giving it good water solubility. Very importantly, HPBCDs are approved for use in parenteral applications of small molecule drugs. The applications of HPBCDs to stabilize protein formulations have been reported in several publications. However, if you notice, few studies were performed on monoclonal antibodies. There are two main hypotheses on the stabilization mechanism of HPBCD in liquid protein formulations. It is postulated that the cavity of HPBCD can bind or interact with the exposed hydrophobic residues on the protein, shielding against hydrophobic interactions between protein molecules, which can cause aggregations. Due to the surface activity of HPBCD, it is also postulated that HPBCD can behave like surfactant, displacing protein from air-water interface, thus inhibiting surface-induced aggregations. However, relatively few mechanistic studies on HPBCD in protein formulations were available. The exact stabilization mechanism is still an area which requires further investigations. As such, our R&D group has embarked on a study to answer the following questions. Firstly, can HPBCD serve as a functional alternative to surfactants in protein formulations? Can it also inhibit heat-induced aggregations? What is the protein stabilization mechanism of HPBCDs? And lastly, what is the chemical stability of HPBCD in relevant formulation conditions? In this work, we have chosen bevacizumab, a monoclonal IgG1 antibody, as the model protein. It is a successful commercially marketed biologic known as Avastin, used to treat a number of types of cancer. Its commercial formulation consists of 25 mg per ml bevacizumab in 50 millimolar phosphate buffer at pH 6.2. It also has 60 mg per ml of trehalose as a stabilizer and 0.04% polysorbate 20 as a surfactant. In our studies, we have attempted to replace these two excipients with HPBCD and study the effect on protein aggregations. Two Rocket biopharma grade HPBCD were used, Kleptos HPV and Kleptos HP. They differ in the degree of substitution, with Kleptos HP being slightly more substituted. We investigated the effect of 2-HPBCD on bevacizumab aggregation during stability studies. Bevacizumab was formulated at 25 mg per ml in 50 millimolar phosphate buffer with varying concentration of HPBCD 50, 100 and 150 millimolar, and compared with control formulation buffer only, and commercial formulation denoted by CF. All formulations were prepared in triplicates. The formulation were then subjected to shaking stress at 1000 rpm and heat stress at 40 degrees Celsius for specific time points, and their aggregation levels were then evaluated using size exclusion chromatography. After agitation, the aggregate levels in the formulation were evaluated. As seen from this graph, we can calculate the rate of aggregation after 24 hours of agitations and perform a t-test. Formulation containing kleptose HP and HPB exhibited either comparable or significantly lower rate of aggregation as compared to commercial formulations. The aggregate levels in the heat stress formulation were also evaluated at 1, 2, and 4 weeks. Presence of kleptose HP and HPB was able to reduce the rate of aggregations of bevacizumab by half or more as compared to the commercial formulation containing trehalose. The aggregation reduction property also appeared to be kleptose concentration dependent. For example, at week 1, the rate of aggregation was 1.8% per week for the commercial formulation. The presence of kleptose was able to reduce this rate to between 0.9 and 0.5% per week. This will potentially help to extend the shelf life of bevacizumab significantly. The potential use of HPB-CD in bevacizumab formulations had led us to embark on a mechanistic studies. We studied 3 aspects. Firstly, we studied the surface displacement theory by measuring the surface activity of kleptose HP and HPB. We performed the binding affinity of bevacizumab and kleptose HP or HPB using isothermal calorimetry. Lastly, we studied the effect of kleptose HPB on the protein-protein interaction of bevacizumab using dynamic light scattering. The static surface activity, or also known as surface tension at equilibrium, of kleptose HP and HPB was measured. We observed that both kleptose HP and HPB exhibit surface reduction properties. However, when compared to polysorbate 20 and 80, they present much weaker surface activity. This may imply that surface property is not the sole reason for reducing protein aggregation. Furthermore, it is important to note that kleptose HP and HPB do not possess a critical micellar concentration, or CMC, unlike surfactants. Their surface tension decreases with increasing concentrations when plotted against the log of concentration. During agitation, a constant renewal of the air-water interface takes place. Instead of just measuring the surface tension at equilibrium, monitoring the dynamic surface tension immediately after surface formation is also important. Accordingly, the maximum bubble pressure method was used to measure surface tension over a surface lifetime. By itself, bevacizumab, the black line, is surface active as reflected by the decrease of the surface tension, and it will absorb at the air-water interface, which means that it might be prone to surface-induced aggregation. At newly formed air-water interfaces, as seen from the initial part of the graph, slower equilibrium kinetics of polysorbate 20 versus kleptose HP and HPB was observed. 100 millimolar of kleptose HP and HPB actually led to a lower surface tension than 0.04% polysorbate 20 at the start of the surface formation. This could be a possible reason for HPB-CD to stabilize bevacizumab as effectively or better compared to polysorbate 20, despite the lower surface activity at equilibrium. We studied the interactions between kleptose HPB and bevacizumab using isothermal calorimetry. The y-axis represents the heat signal after injection of the ligands into the sample cell. The blue line in the graph represents titration of 5 millimolar kleptose HPB into the bevacizumab solution. Very low heat signal was actually observed, similar to titration into the buffer, as shown by the pink line here. This indicates that there was almost no binding between bevacizumab and kleptose HPB. Similar observation was also noted on kleptose HP. The effect of kleptose HPB on the protein-protein interactions of bevacizumab was also studied using dynamic light scattering. We first measured the hydrodynamic diameter of bevacizumab at different protein concentrations. It was then converted to the diffusion coefficient by the Stokes-Einstein equation. The dissociation constant Kd can be determined by the linear fit of the measured diffusion coefficient as a function of concentration, as seen by this black line. Different concentration of kleptose HPB, 25, 50, and 100 millimolar, were then added to the bevacizumab solutions to study the effect of kleptose HPB on the Kd value. The Kd values were found to be less negative in the presence of kleptose HPB, indicating a rise in net repulsive forces between the protein molecules. We postulated that the presence of HPB-CD may lead to a change in intermolecular forces between the protein molecules, resulting in a reduction in protein-protein interaction and hence a propensity for protein aggregations. In summary, we observed that there's no binding between kleptos-HP and HPB with bevacizumab. Although kleptos-HP and HPB exhibit weak surface activity, it might not be the main reason in reducing protein aggregation. The reduction in protein-protein interaction may largely account for the stabilizing effect of HPB-CD on bevacizumab during agitation and thermal stresses. Lastly, an accelerated stability study on kleptos-HP and HPB was conducted. 200 millimolar kleptos-HP or HPB at different pH condition and salt content were analyzed using petrol-oxy oxidation stability tester. The samples were subjected to 40 degrees Celsius and at an oxygen partial pressure of 700 kPa for 12 hours. The percent oxygen decrease indicate a poor consumption of oxygen, meaning no reactivity was observed in any of the samples. Subsequently, the hydroxypropyl substitution on HPB-CD was measured using MMR, and the reducing sugar titration was performed to determine any hydrolysis of the HPB-CD. All results indicate that kleptos-HP and HPB were stable under elevated temperature and oxidative stresses. So what conclusion can we draw from our studies? Firstly, kleptos-HP and HPB presents weaker surface activity compared to polysorbates, implying that surface property is not the sole reason for reducing protein aggregations. ITC data indicate no evidence of strong interaction between kleptos-HP, HPB, and bevacizumab. But protein-protein association is found to be weakened by kleptos-HPB. Lastly, kleptos-HP and HPB do not undergo degradation under elevated temperature and oxidative stresses. This study has offered a deeper understanding of the mechanistic role of kleptos-HP and HPB in bevacizumab formulations. It has also illustrated that kleptos-HP and HPB are viable options for reducing aggregation of bevacizumab, and therefore extending its shelf life. Thank you very much.